Hello, everyone. My name is Paulo Henrique Olivieri Jr., and I'm here to present my work entitled The Influence of Glycocalyx and Protein Corona Interactions on Silica Nanoparticle Uptake. Engineered nanoparticles have remarkable potential for diverse biomedical applications. Over the last few decades, numerous studies have focused on characterizing and understanding these biointeractions of NP with cells, especially factors that affect cellular internalization of particles. However, predicting or explaining these outcomes remains challenging due to the complex nature of NP cells interactions and the endocytotic processes. On the one hand, the formation of an absorbed protein corona around nanoparticles is a critical factor that influences these interactions and endocytosis. In addition, when these nanoparticles approach the cell surface, they first encounter the glycocalyx, a negatively charged glycosaminoglycan or GAG-rich layer attached to the plasma membrane. Recent studies have suggested that the glycocalyx may play an important role in regulating endocytosis, as it can either hinder or facilitate the processes, depending on the NP and the glycocalyx characteristics. However, these interactions between the protein corona and the glycocalyx and their impact on the NP uptake remain poorly understood. Therefore, the main goal of this project is to observe the effect of the GAGs along with different protein corona models in silica nanoparticle endocytosis. To sum up, all the experiments were performed in the same cell type, which is a CHO cell model, that is a Chinese hamster ovary cells. We utilized 3 different artificial coronas, one made by BSA or bovine albumin, one by a modified BSA to be positively charged, and one with complete FBS or fetal bovine serum. All experiments were done up to 4 hours of incubation with the nanoparticles, with or without the treatments. And all of our models were either the CHO cell model that is wild type or mutant. We also performed an enzymatic GAG cleavage, the chemical GAB inhibition, and the heparin competition assay. All of those experiments are done to try to do different approaches to see whether the GAGs help or make it difficult for the nanoparticles to get internalized by the cells. So the results that we found were quite intriguing. First of all, in our cell, by using our cell model, we observed that all 3 coronas, especially the BSA positive, get their uptake decrease when we remove the GAGs, showing that the GAGs might help maybe as a receptor or maybe influences the contact with the nanoparticles, increasing the uptake. Second, by removing specific GAGs from the cell surface, we can try to observe if we see a decrease specifically with one of the GAGs. In this case, we observed that especially when we remove heparin sulfate, we see a decrease in uptake of the nanoparticles, as well as when we remove all the GAGs as well. Third, we made use of chemical inhibitors to decrease the GAG expression. One of them being sodium chlorate, another being a pharmacological inhibitor called mitoxantrone. And again, we see a decrease in uptake only this time with the BSA and the BSA+. Lastly, we try to observe the effect of heparin presence in the nanoparticle endocytosis. In this case, we're trying to increase the number of GAGs in the surface, try to modulate the competition to see whether this heparin presence increases or decreases the uptake. In this case, when we increase the concentration of heparin, we also decrease, the uptake of these nanoparticles. So basically, in conclusion, we observed the effect of GAGs in increasing the uptake of corona-coded nanoparticles was observed in CHO cells by 4 different approaches. But to generalize this hypothesis to most cell types, other experiments will be performed to other cells to either robust or deny our hypothesis that the GAG helps the uptake of nanoparticles. For now, we are also doing experiments with HeLa cells, and we discovered the same trend that we saw with the CHO cells. We also plan on doing other experiments with endothelial cells, that being important to understand, because of the endothelial cells being the first barrier that the nanoparticles encounter to go deeper in the body. And we're also doing the same experiments in another cancer cell model. Thank you so much for your time, and have a nice day.